but when they continue variation after domestication, it's called secondary center of domestication. These are the candoles. This is the father of crop evolution, and here is Vavilov, the person who actually proposed that domestication started in 8 major centers around the world, 2 in the Americas, North Africa and East Africa, and then the Mediterranean, the Fertile Crescent, Middle Asia, South Asia, the Far South Asia. These are the 8 centers of domestication. And these 2 plates show what plants and animals were domesticated, because the domestication plants and animals were together. When you plant plants, then you need animals to live. These are the 8 centers and the examples of what plants and animals. For example, barley and wheat, the cereals were in the Fertile Crescent, but cotton and maize were in the Americas. And here is the timeline where and when it happens as well. And when domestication occurs, there is some characters or traits associated with domestication. What benefits domestications have given the crops at the early stage of their development? More yield, of course, of desirable parts. Gigantism, the size of the plants became bigger, and the seeds became bigger, and the fruits became bigger. A uniform ripening of fruits and seeds, uniform seed and fruit size. More growth, increased apical dominance, means a continued growth upright by the plants. Non-shattering of seeds. When the seeds are ripe, they are not shattered from the main plants. And this makes a harvest easier, improved quality, no lower toxic substances, for example, increased protein, increased oil, increased sugar, and also improve the flavor and storage. We still breed for these more sugar, more oil, more flavor, more storage ability. These are still major topics in agriculture. We also have reduced branching. We have less reduction of seed dispersion. We loss or reduction of seed dormancy. And of course, this is more science than public information, but these are the major domestication syndrome. It's called syndrome because they occur together at the same time. It's very close time as well. What much domestication of plants? Only 2,000 plant species have been grown for human food. That is less than 1% of all plant species. Today, even concentration on 150 species are commercially grown for food, not including spices, for example. But if we look even closer, we will observe that we will see that about 50 very productive species supply most all of our caloric needs. Particularly cereals, for example, wheat, barley, maize. These are cereals. Legumes are major part, of course, but the benefits of domestication was that happens 10,000 years ago, or started 10,000 years ago. Agriculture began. The world total human population was about 5 million at that time. Now, every person at that time have 25 square kilometers. Today, the human population is 8 billion people with a density of 28 people per square meter. So how much is the world that changed? From 5 million to 8 billion people, and the share of each person have actually gone very smooth. This is the people how domestication started. There's some practice that we still use in our development, but these are very important. In this concern, we mentioned that crops are not only grow where they were domesticated, but crops are international travelers. For example, cotton and the maize come to the old world from the Americans, the Americas, and wheat and barley went all over the world from the Fertile Crescent, and rice came from Asia. That is importance of traveling by crops. This is an example of how barley actually traveled from the Fertile Crescent to China and to Ethiopia. Domestication also involves what they call genetic bottleneck, means the scientists or agriculturalists actually choose the genes that make production more. So they concentrate on the most benefits of the most productive genes at the cost of other genes. So the wild relatives has many genes. Some of them were lost at the early domestication, and more are lost in the modern varieties. That's why genetic resources are important, because when some of these lost characters are required, we go back to the wild species or the genetic resources to get these genes back to the crop we cultivate now. Domestication involves genetic bottleneck. Only few genes are actively selected, and large shifts in the phenotype means the external appearance of the plants. Methods of crop improvement. After domestication, we still improve crops. We still look for more food because the human population is growing faster. We need also crop production to grow faster than the human population. We use natural selection, we use hybridization, we use breeding by both natural selection and hybridization. But recently, biotechnology came into being, but through tissue culture and through genetic engineering. And these, with the time, how the maize was developed, how the oil content of the maize kernels increased with time, from 0 to 60. Here are some photos of the crops before domestication and after the wild maize and the current maize, the wild rice and the current rice, the wild wheat and the current wheat, the wild barley, the current barley. Of course, if you remember the domestication syndromes, the 10 benefits of improving the production, we will actually find them photographed in this simple photo for these important cereals. Another technology used is tissue culture, genetic engineering, smart agriculture is the future. And here we use molecular tools in genetic resources to develop more genetic resources. We use diversity analysis of germplasm, as we said, like the evolutionary relationships, development of core collections, gene flow studies. We use DNA fingerprinting to identify germplasm and the genetic priority. We are concerned with the genetic priority of the crops and identification of duplicates, gene discovery, association mapping, using techniques called association mapping, and the allele mining. We mine for genes and the types of genes that would be beneficial. And of course, we also look for trait-specific germplasm, identify the traits and validate them. These are what they call the forward phenomics. We start from the plants, and we look for the genes, or start with the gene and look for the plant characters. There is forward phenomics. If we started that with the traits, we are looking for the characters we are looking for, and look for genes, or start by identifying genes and try to use to make genes more efficient for producing more useful traits. International concern about, of course, there is some international concern about genetic resources. The South have plant genetic resources, gene-rich countries, developing countries mostly, and in the North have improved the cultivars. They are gene-poor countries, but they are developed. So because with their technologies, they take the genes and they develop more productive cultivars. Then they actually sell the South the expensive cultivars because they have made some knowledge. That's why the developing countries in Africa and Asia should make more efforts to develop knowledge about the crops and to develop knowledge about utilization of natural resources. And these make the use of these natural resources for the business, for the usefulness of the population, for the welfare of their societies. Here is the Thomas Malthus theory of population, because if the human population is growing very fast and the food production is not sufficient, then human population exponentially, while the food production grows exponentially, there was some called Malthusian catastrophe. And this will make problems with wars and famines and poverty and everything. The other theory is called post-Rome theory, responding to the Club of Rome, because in the 1960s, when there was a problem in food supply, the FAO, the Food and Agriculture Organization, have arranged some efforts, international cooperation, to increase food production so we can make food production always ahead of human population increase. And this is called the post-Rome theory, responding to the Club of Rome. An international treaty, of course, they made the international treaty to assist countries, particularly the developing world, in need of plant genetic resources conservation and strengthening their links to users, and to build international collaboration in crop and geographic areas, and to develop strategies and technologies for the conservation of plant genetic resources, and to provide more information and services about the. Here are conservation of plant genetic resources. There are two theories, two methods to conserve genetic resources. One is called the species-oriented approach. It depends on the species, on the crop itself, and you can keep it in conventional, orthodox as seeds, or to keep it in vitro using tissue culture or pollen grains or cell suspension, or even the genes called DNA. And of course, you can use it in cold rooms, and of course also in DNA libraries, and you can still have field gene banks and like botanic gardens and the greenhouses. The other approach is called ecosystem-oriented approach, and this actually not only conserves a single crop, but conserves the ecosystem as a whole. And with conserving the ecosystem, we actually conserve habitat ecosystem and conserve population, and these are available as bioreserves and heritage site, wildlife sanctuaries, and farmers field and the tribal areas. These are the conservation of genetic resources. International cooperation actually has led to establishment of centers around the world, one in Nairobi, one in Syria, one in Sri Lanka, in India. So these were actually establishment made by the Food and Agriculture Organization to make international cooperation possible, and this actually has led to more improvement with regard to acquisition or collection of genetic resources, which these are the management ways to cooperate on collection and acquisition, to cooperate on regeneration, to cooperate in characterization, evaluation, and documentation, and also in distribution. Actually, genetic resources are very important because it does not only concern improvement of food production, but also with conservation of genetic traditional knowledge. The human being has developed the knowledge with their civilization for 10,000 years. And these traditional knowledge are parts of human life, not only in developed countries, but mostly in the developing countries. So there is some concern that we also need to conserve the traditional knowledge, and that may be oral and rural in nature. that may be dynamic and based on innovation and adaptation, that may be concerned with communities, that location and the culture specific, decision making on survival and strategies, and also not systematically documented, and concerns critical issues of life. These are the traditional knowledge. Back to the increase in annual production of cereals is a major topic. Actually, there was a very famous scientist called Norman Borlaug in the 1960s who developed the theory of increasing food production by enhancing the plants to produce food rather than vegetation. So to reduce the energy spent by plants on production of food parts, not the leaves and other parts that's not producing food. And this actually led to major increase in the major crops like here cereals, maize, rice, wheat, barley, sorghum, millet, and oats. If you compare 1961 with the 2019 and 2020, they will find almost five-fold increase in maize, almost threefold in rice, almost threefold in wheat and barley and other crops. This was very important step forward in producing in improving crop production. Climate change, of course, we all know climate change as defined by the United Nations is a long time shift in temperature and weather pattern that may be natural or through variation in solar cycle. As I said in the beginning, I will not focus on the types of climate change and as such, but I will just go through some slides about climate change. And climate change is a long-term process, while weather is the state of atmosphere, especially temperature or humidity at a short scale, even hourly changes or daily changes. These are famous slides showing the 10 indicators of climate change. Some people consider more. Some people classify causes and effects of climate change. This would be available in this slide. And here are the major contributions to the emissions of the greenhouse gases. And greenhouse gases, of course, are the major sources or the major causes of increased temperature, and the increased temperature is the major cause of climate change. Carbon dioxide is of course the leading, but methane, nitrous acid are major contribution. And here are the same industry, transportation, agriculture, electricity, and heat are the major sources of contribution to climate change. Here is the association, close association between carbon dioxide and temperature, and here carbon dioxide, global temperature, and sea level, and then back to the genetic resources in adapting to climate change. Climate change will change rainfall, loss of soil fertility, cause fluctuation, temperature, and light, may increase evaporation, transpiration. Transpiration, by the way, is the loss of water in the form of vapor from plants. Plants do transpire water because of the sort of vapor because they need water in their bodies to be moving. This actually controls the temperature of the plants. It's a very important, it's a loss of water, but it's very important for the life of plants. That's transpiration. Biological stresses, increased pollution, declining in biodiversity. These are 7 severe causes of climate change that may actually affect the genetic resources. And under genetic resources, of course, breeding procedures, we need more breeding procedures to accommodate and the screening for environmental stresses. Responsive genes on crop genetic resources is a major step forward in selection of tolerant traits or tolerant characters of plants, of crops. Improvement in both water resources and their utilization are needed to elevate final yield under water deficiency. Under climate change, of course, we will Under climate change, of course, we will need more improvement of water resources, more improvement to utilization of water, because we will be underwater deficit conditions. Many pathways are associated with genes controlling morphological, that is external appearance of plants, physiological, that's the reactions in the plant bodies, responsive, and determine plant performance, how to improve plant performance under stress, under environmental stresses at various stages of plant growth. This is a very challenge of plant scientists to make a genetic resource and the crops able to be resilient to climate change. And the plants have actually learned this across their long life on Earth. And they do this by three mechanisms: the escape, a short vegetative period, early flowering, early maturation. The life cycle of the plant is too short because they are not sure that the water is available, so they finish their life cycle very soon, very early. Tolerance, they live, but they manufacture some compounds that make them able to live under stress. These, like antioxidant biosynthesis, osmotic adjustment, improved water content, photosynthetic recovery. These are mechanisms made by plants themselves to make themselves tolerant to the stress conditions. Under stress conditions, the third methods is avoidance, is to avoid stress. And the avoidance may be possible by deepening roots down in the soil, so searching for water in the deep soil, or to reduce the loss of water by transpiration. That's the stomatal activity. The stomata are openings in the plant leaves that controls the loss of water. For this, stomatal activity has to be more efficient. Leaf shape change to make leaves smaller so they can lose less water, and transpiration be lower, and of course they can delay their growth, so they can need less energy. These under drought stress, biochemical responses and the molecular responses and the physiological responses. I will not go into details about the mechanisms of biochemical response or molecular response or physiological response, because that would be difficult for the public to understand. But at least we know that molecular response happens at the means by which the plants adapt themselves, and the physiological response, the reactions that control stomatal activity or gas exchange or osmotic adjustment or water nutrient uptake. These are mechanisms. Of course, it's easy for botanists to understand this, but I will just pass this and just ask you to remember that under drought stress, there is biochemical response, molecular response, physiological response. And plants also under stress, they have different ways and means to tolerate drought or cold, not only heat. They may have drought, meaning less water, salty, saline soil. They may face heat, higher temperature. They may face cold or freezing environment. So the different plants will develop different means to live and adapt to their environment. And when we discuss about this or talk about this, we need to understand response to heat stress, for example, at the rooting to anthesis. From the plants are pollen to anthesis. Anthesis means to flowering and fertilization and the green thing. There is different plant stages that respond differently to the stress conditions. So the botanist and agriculturalist need to understand how the plants will respond in the early stages of life or the middle stage of their lives or after harvesting. These are important topics that the scientists and botanists and agriculturists must address to produce tolerant crops to climate change. Here is a schematic workflow to get improved varieties, starting from landrace collection and improved varieties, landrace information, landrace characterization, phenotyping to make databases about phenotyping, genotyping databases about genes, and from understanding the phenotype and the genes. The phenotypes is done by phenotyping, and genes are made understood by genotyping. We can then make genomic selection, genome-wide molecular markers. These are more developed methods of selection of crops or new cultivars of crops that are more tolerant to climate change. The action plan is to do experimental design, measurement of stress responsive traits, this phenotyping, evaluation of yield parameters under environmental stress, understand, utilize transcription factors. Transcription factors are new means of regulating abiotic stress and signaling pathways. These are sometimes signaling the signal perception, signal transduction, and abiotic response. These are important ways to understand the response of plants to stress. Genomics and environmental stresses, phenotyping and genome database, they are very simple methods. Here is stress and the controlled genotypes. You grow genotypes here, you record the stress responsive traits, gene expression data, genome data, and from these you go to the web access and to go to databases and analyze your data and select what traits to improve and what genes are behind these traits. The last 2 years, we have done some work on the screening for stress-tolerant maize germplasm, and the experimental results of the screening in maize germplasm for valuable traits that might include abiotic stress for bleeding potential. This was published in Photosynthetica as a Photosynthetica is a very famous journal that published plants related to the process of photosynthesis. Photosynthesis means the capability of plants to transform carbon dioxide in the air into carbohydrates, into sugars in the plants. This is a very essential, crucial character of plants. They actually use carbon dioxide, which is actually increase Earth's temperature, into sugars for the consumptions of humans and animals. That's why the plants are known as producers in the ecosystem. Plants are the producers of life on Earth. That's why we are concerned about climate change and concerned with their action on plants. Here we tried some simulation of drought stress, and here some doses, and here we've measured some traits responsive, like shoot length and root fresh weight, and root length and root fresh weight, and root dry weight. These are traits, and then we analyze the data, and we actually, these are the germplasm we used from different parts of the world. This was done in cooperation between Helwan University in Egypt and the German Gene Bank. I've been in cooperation with the German Gene Bank for about last 4 years, and we will be going continuing cooperation this summer. I will be going next July for 3 months to work this time on barley. We started with maize and published these results, and we calculated what they call DTI, drought tolerant index. With this index, we compare different landrace different genetic resources from the same crop and determine which form or which germplasm is more tolerant to drought than others. And another paper about maize as well was to this global collection to connect the ability to adapt to climate change in different parts of the world with the fingerprinting. Fingerprinting, this is the DNA fingerprinting of different germplasm of maize from around the world. And with this, we measure the genetic relatedness between different forms and how are close to each other and how can design experiment for future screening of more adaptive forms of cultivars. This method called cluster analysis. Cluster means clustering the very close forms together. The closer they are, they are more genetically similar. And when we compare these together, we select which trait can we use for production of new crops. Lastly, we made some comparative analysis of the drought stress response of maize genotypes using chlorophyll fluorescence. Chlorophyll fluorescence is a very important feature of plants. When plants do photosynthesis, which I just mentioned that they could transform carbon dioxide into sugars in plants, they do this with the aid of chlorophyll. Chlorophyll is the green light of plants. And by the way, it's very similar to the blood in a human being, with the replacing the atom ferrous with the atom magnesium in chlorophyll. So with this chlorophyll, when the chlorophyll receives light, they fluoresce. And this fluorescence, if measured, actually indicates how plants can be resilient to climate stress. And with this, actually, this is the control, and here is the simulated drought, and here is the measurements of chlorophyll fluorescence. There's some curves and some cluster trees, which are difficult to explain to non-professional botanists, but these are the forms of proteins, the difference in proteins, special types of proteins. And with this, we go to what we call proteomics. Proteomics means trying to find what changes in the protein under stress. And here we calculated what they called, this is a very famous picture of proteomics. And these are the proteins that change in tolerant maize cultivar from Libya, and these more in less tolerant maize from Italy, because the Libyan forms of maize were more tolerant, were more tolerant than the maize from Italy. This was proved by the analysis of proteins in the plants under stress in both genotypes. Crop improvement by integrating multi-omics techniques. Omics, of course, I mentioned omics is the fashion of the day to do genes. That means you are working genomics. When you do, you work with the mRNAs. This is a messenger RNAs, and RNA is a faithful copy of DNA genes. When the genes work, actually, genes are in DNA. And they work by sending the faithful copies of themselves, called messenger RNA. And these messenger RNA are transcribed from DNA. When we are working with mRNA, we are actually working with transcriptomics. So working genes is genomics, working with messenger RNA that transfers the message of genes is transcriptomics. And this message is translated into proteins. These are called the proteomics. And proteins actually as either enzymes called functional proteins, which control the metabolism, control the reactions in the living organisms, not only in plants, but also in all living forms. These are called metabolomics, the chains of reactions in the life forms, in different life forms, which came from genes through messenger RNAs to proteins to in different reactions to give us at the end the phenotype, the performance of the plants depend on genes, but our genes are actually work under environment. And with climate change, the environment will be more harsh. And that's why we need to make genes more efficient and they make through producing new message and new proteins and new reactions that would make plants more resilient to climate change. And the last slide of my talk, in using the potential of genetic resources, improving agricultural production or crop productivity, three major actions must be taken into consideration. The first is to avoid actions that may destroy biodiversity. And biodiversity, of course, a very common name. It's very common to the public, like climate change, because people kept talking also about biodiversity. And since 1992, the world has the Treaty of Biodiversity. So we should not destroy biodiversity or harm the health of agricultural ecosystem. For example, we should not use pesticides that may kill pollinators and harm other forms of life. We also need to improve knowledge and methods for conservation and use of crop wild relatives to expand genetic resources and to intensify efforts to use biodiversity as a reservoir of genes and the traits for improving the genetic resources and the development of new cultivars. And also we are in need for new policies to support such an approach to avoid useless actions. And with this, I end my talk and thank you for patience and listening.
Thank you so much, Professor Abdelfattah. That was really some good stuff. And maybe, colleagues, if you missed out something, I would love us maybe to always, chip into our inbox and ask those questions. But for now, Doctor, maybe if I can just get you some of the questions that our colleagues were asking. Mekanon from...
Should I stop the screen now?
Yes, you can. Yes, of course, you can stop sharing. Mekanon from, this is a journalist from Ethiopia, is asking whether you would give some comparative analysis of genetic resources of countries around Egypt and the East African region. What are some of those, comparative analysis that you think?
Comparative analysis of genetic resources?
Yeah. Yes.
Yeah, of course, we need different, if we have different forms of genetic resources, we need to actually calculate the performance of each form, and these are called comparative analysis. And to compare, we need to use the same methods. So we should not apply different methods for different situations. I think you are muted. Because
Doctor, Professor, we have another question.
Yes.
And also, so the question is, for the people that criticize this kind of thing, the people that criticize these GMOs, they come in and say that genetic engineering, believe that genetic engineering is likely to cause extinction of some traditional crops. And also, there is the worry that these genetic, produced plants are less resistant to pests and diseases. What is your take on that?
Yes, this is a very common question for the last 30 years or so, since the development of genetic engineering in the 70s and the production of the first improved plant resistant to herbicides and resistance to insecticides. Actually, genetic engineering, like any other invention, have positive sides and the negative sides. If we use genetic engineering, we have to observe what they call the biosafety measurements to make sure this gene that was introduced from a foreign origin is integrated in the genotype of the species and will not be harmed. Of course, there is some characters that should not be engineered, like wheat, for example. Many crops have been engineered, including maize, but maize is actually engineered for as a fodder, not for eating, or for oil production, not for food. So with food, we need to be careful, but for other purpose, it is okay. In the recent years, I must say, genetic engineering has been less attractive even for the geneticists, even for the scientists, because it's more costly. It likes more knowledge. But now new alternative called genetic editing, gene editing. Gene editing means gene repair. You can repair the original gene of the plant to produce more. So you do not actually introduce foreign genes to the crop. You are dealing with the genes of the same crop. And this is usual because plants have changed their genes with a long history of evolution. The plants today do not have the genes that they do have 10,000 years ago, but genes have been lost and regained by natural selection, by hybridization. And if we can have a safe way to improve genetic performance by genetic resources, that would be a step forward.
Professor, we have just two more questions from the discussion that we have had. Then maybe I can also invite the journalists to ask. So I'm asking myself, how are developing countries, how is this likely to enhance the national income of, developing countries like Uganda, Rwanda, Burundi, Tanzania? How is this going to help them?
Most countries are parties to international agreements like the biodiversity, like the genetic resources exchange, and most countries now have gene banks. And these genetic resources, not only of plants, but also of microorganisms and other sources of natural resources are kept in gene banks. With plants are kept in gene bank, and every country has a gene bank that collect its genetic resources and they keep and they document and they manage at the national level. And if we are rich in genetic resources, you can actually exchange these for more genetic resources. So to answer your question, how to do this, it's done by gene banks. And the gene banks have scientists, agricultural scientists, botanists, and geneticists, and people qualified to collect, to characterize, to document, to manage genetic resources.
Thank you so much, Professor. I will just ask you one more time. This is a question coming in from Kenya, and someone is asking, so they are concerned. From what they have heard is that these genetic plants are the sources of cancer. Is it true, or this is just bad talk and hearsay?
No, that's not true. Genetic plants, of course, plants there are some toxic plants. There is some actually the toxin produced in plants may be useful for in medicine. So toxicity is a harmful, but for our enemies it is our benefit. Anyway, plants actually does not concern, it does not cause any harm in the future. Plants are very friendly to the human being. Plants are the producers of food on earth, and it's unlikely that they can cause cancer. Cancer is caused by many other things, but not including that. Actually, probably the therapy of cancer maybe come from plants. And many plants, by the way, produce antioxidants, which make people more resistant to cancer.
I think this has been really interesting and eye-opening, Professor Abdelfattah. I think we will need to have you more to have, some of these questions raised. There are so many questions from friends, and we are already 5 minutes into the hour that, we wanted to stop. But we still have 15 minutes because we started a little bit late. But maybe for now, I could give another opportunity, maybe to 2 or I don't know, 3 or 4 journalists to answer. I mean to ask some questions. I see here 2 hands raised, and maybe my colleague who raised first was Christopher. Christopher, let me ask you to unmute and maybe you ask your question. Thank you so much.
Yeah, good afternoon, everyone. I'm Christopher here, Kampala, Uganda. I'm a biotech journalist. I've been reporting on GM technology for the last 13 years. I believe it's the current technology of the current generation and the future, because if we are using it in the production of medicine, I don't see any problem why we can't use it in trying to solve challenges of food production, like low nutrition value in bananas or plants that are struggling with water. But my question, Professor, is I've seen the graph. I've seen the graph, the increase in production among the different crops: wheat, maize, sorghum, and millet. So I'm concerned. Why is it that we see a triple increase in maize? We see a huge increase in wheat, but there is very little increase in millet and sorghum. That's my question.
It's very interesting question from Africans, because millet and sorghum are mainly grown in Africa. But maize is a worldwide crop, and wheat is a worldwide crop. So if the world has more interest in sorghum and millet, their production will be increased because they will give more efforts and probably more knowledge. But I assume the millet and sorghum are crops for the future, because they are less studied, they are less understood, but their productivity is very high, and probably their nutritional value is high. So they need more efforts. But the reason why they are not so developed, because they are more cultivated in the Sahara area of Africa or in African countries on the whole.
Okay,
it's
Professor.
not bad they grow in Africa, but I think Africans should put more efforts to develop more productive cultivars on this. By the way, I have a Sudanese student working on sorghum. Yeah.
Okay. Thank you so much, Professor. I will ask Wafa. Wafa, I don't know if I'm saying your name correctly. Wafa Ama. You can go ahead and ask your question.
Yes, thank you, dear organizers, for this informative lecture as well. Great, you are inviting a great professor, our Professor Dr. Abdelfattah. He is covering the point in a very interesting way. But I will maybe replay, or I am on the side of Dr. Abdelfattah, because you are asking about how can Africa enrich their crop productivity and germplasm productivity or gene productivity. I guess Dr. Abdelfattah answered in his recommendation that we should interest paid interest and effort for the wild crop relatives in Africa, as well as the underutilized crops. Why I am saying this? Because of the 2 issues. However, Africa suffering from threats of different waves of hunger and drought. Africa hosting a huge, not less than 5,000 species and land races underutilized and all of them are qualified to be domesticated in the future if more resource paid to this issue to enrich or to enhance this its crop productivity. So we should pay a great effort to our genetic resources, because Africa deserves this. If we see just through have a what's called going through Europe, you find a association of wild crop relatives, and all the crop relatives in Europe, its sites recognized, its position recognized, and there is a collection of germplasm deposited in gene banks, and they're trying to improve wild crop relative. Why? Because most of the domesticated crops in the future, its productivity by the coming 20 years will be decreased at least 25%. So we should secure our food resources, especially in Africa, because we have our resources, and we have our resources. Maybe it will cover all the world as for its the future food all over the world. So I am on the side of Dr. Abdelfattah in his recommendation that we should seek paid interest, paid attention to our underutilized and wild crop relative. Thank you for this interesting lecture. Thank you. Good luck.
Thank you so much. Right now, friends, I know you have a lot of questions to ask Professor Abdelfattah, but I think also Wafa is an expert. I don't know if I'm correct. Madam Wafa, are you also an expert in this kind of thing?
Yeah. I am a biodiversity expert and IUCN member in wild crop relative section.
Okay, and that's very nice. And for those of us who would love to link up with Professor Abdelfattah after this for more information, please look into the message section. We have put his account, his Nilewell account. You can always go there and reach him. I will ask Professor Abdelfattah to also help Professor Amma to open up another link. And also remember that we have very interesting projects or services. One of them is the WhatsApp chatbot. The number that has been posted in our message box, you can easily just say hi and you will see various things that might interest you. That could be our weekly or monthly updates on what we are doing. It could be stories and it could be even opportunities. So please try to check it out. But for now, I want us to get into a section where we will be winning some stuff. It's been a long day of listening to great stuff, but also we want you to take home some prizes. I will invite my colleague, Christopher, to come in, and he's going to be asking a few questions that basically are around what Professor Abdelfattah has been basically communicating. And from there, we will pick a winner. And the winner will have a very big prize. Very big prize.
What about me? Would you send the name?
Yeah, if you win a prize for the audience, what about the
Professor, it will be cheating. It will be
Uh,
cheating, Professor.
So the professor is not allowed to send any cheats to anyone. The professor should be neutral because we want to see whether actually the class has been paying attention. So with those short words, maybe I can call Principal Chris to take over. Thank you so much.
All
Chris,
right.
over to you, please.
Thank you, my brother Andrew. Welcome everyone to our monthly Science Wednesday. I'm so happy to be here with all of you. The last two we've had have been interesting, have been fun. So this time around, we're also going to have a few trivia questions, not so difficult. But from what Professor has been sharing and teaching us, giving us a lecture on, just to jog our memories and see if we actually paid attention. They're not very difficult. All right. So Andrew, you will help me keep score who shares the correct answer in the message. Yeah. All right.
And Judge Curity, we all need.
All right. Thank you.
Yeah, sure.
All right, guys. So we are going to start with number one, and it's very easy. It's very easy. Dash is a robust tool aiming to detect quantitative traits.
We dropped you,
Of
Chris.
large-
Maybe you can repeat again.
Oh, all right. What about now? Do you get me?
Yes, you're now clear.
Okay. I was saying DASH is a robust tool aiming to detect quantitative traits of critical agronomic traits by combining genetic, geotypic and phenotypic data. Even me, yeah. One more time. DASH is a robust tool aimed to detect quantitative traits of critical agronomic traits by combining genotypic and phenotypic data. Just write the answer you can remember
Yes.
from Professor.
We have Christopher Benandan, and he's saying genetic engineering. I think he was the first, Christopher. That's the journalist from Uganda who has been covering this topic for over 14 years. That's nice. Thank you so much. And of course, Jonathan, we also see your answer. It is correct. Curity, you say genetics. I think Curity is part of the judges. Maybe he was trying to cheat for someone, and maybe the message went to everyone. So, yes, Emmanuel, we see you. Genetic engineering, beautiful stuff. Let's go to the second question.
All right, number 2. Name the categories of genetic resources. Name the categories of genetic resources. I'll give you a hint. There are 4.
Okay. We have K. Emmanuel. K. Emmanuel says we have land, land rest,
what?
then we have
Yes.
wild space, land rest, and then wild space. So he gives us 2. We have Christopher comes back and he says foundational seed. I don't know whether Professor that's right.
No. That's not right.
In name of professors.
We want 4.
Yeah.
Name the 4.
Okay.
Name the categories of genetic
4?
resources. Yeah.
Kay has given us, okay, so we have a very, okay, we have one called Tarek. Tarek, did you just copy paste this from somewhere?
No.
This was very fast, yeah. Okay, yeah, this is quite interesting, but we thank you so much. I think also Tarek is an expert on this, because from this answer you really see that this is someone who has gone down into this. And of course, Christopher comes back and said, breeder's seed.
Yes.
Yeah, so maybe for now we could, breeder's seed is one of them. So far I see that it is Emmanuel.
Emmanuel was on the right track. He had 2.
Yeah, but
Emmanuel
he gave us
had
only
2.
2. Yes.
You already gave us 2.
Yes.
So do we consider Emmanuel our winner for this round?
We consider Emmanuel, maybe he takes a half of the mark, but yes, of course, he remains the winner because he has. Yeah.
Okay. All right. Let's go to the next.
Emmanuel, you get yourself a half. So he comes back and says, and wild relatives. Is wild relatives part of the answer?
No wild relatives there.
Hey.
No wild relatives.
Okay. So
he gets half
on
a
that.
mark.
So we can go.
Yeah.
Yes, sure.
All right.
So apparently we have Christopher leading, and of course Emmanuel will have a mark. Let's go on.
Okay. How many
species? You still have 3 remaining questions, so please try to.
All right. Well, let's go. How many species have been grown from the domestication of plants? How many species have been grown from the domestication of plants?
Over years, how many are there?
All right, let's go. What are you getting, Andrew?
I'm saying I see here Emma has 2,000 species. I don't know if it is right. And then we have Christopher saying 100. So Emma takes it again. Okay.
Emma is
takes
on
it
fire.
again, 2,000 special. Yeah,
Emma is on fire.
Must
He was
be
very
maybe
attentive.
seated close
This is at the front, eh?
Yeah, oh yeah.
At the front of the lecture room.
Emma is
Yes.
not a bad
Well done.
bencher. Of course, the same answer comes in from Justin Makagala and of course Sia, but we will take, of course, the first correct answer, which came from Emma. We can go on to the second last question, please, if you don't mind.
All right, now we are going to go light now because we've asked some science-driven questions. Let's ask a few general knowledge Nile Basin questions as well. So we shall ask two from this section. So which two countries in the Nile Basin are often referred to as the downstream countries? Which countries in the Nile Basin are often referred to as the downstream countries?
Yes, I see.
Someone has Christopher's won again.
Yeah,
he's the winner here. He has taken that one. Well done.
Christopher says it's Egypt and Sudan. Thank you so
All
much.
Right. One more.
One more.
Let's go one more. One more.
Yes. Yeah, okay.
Which country?
Justin also says sure.
Yeah, which country is the home to the largest percentage of the Nile Basin total population? Which country is the home to the largest percentage of the Nile Basin total population? Again, we have Kai Emma. Mr. Duku has got that one.
Yes. Kai Emma says Egypt. So from what I have here, is this the last one, Chris?
I think that's it for us now. That's it for the trivia.
Yeah.
Thank you so much, everyone. Thank you for participating.
Okay, thank you so much, Chris. Chris, his nickname here.
Yes, it was a pleasure giving this lecture to you and having your cooperation and your fun.
Yeah, maybe doctor, before you leave us, would you love to tell us some more interesting stuff you've been working on? What are some of those interesting things that you're researching? The current research. What are some of those exciting things that your research, airs out? The one that you just did.
The most my most important research in lifetime or currently?
Currently, something that can interest us. Yes.
Country is breeding climate change resilient cereals.
Okay.
Starting with, of course, I started with barley about 20 years ago, but that was about domestication of barley. This photo I showed about traveling of barley from the Fertile Crescent to China.
Yes.
This is the result of one major article published by me in 2000 in cooperation with the Max Planck Institute in Germany. And this has put a new theory of how crops are changed through allele substitution from the Fertile Crescent to new parts of the world. This is the concern. My concern is actually breeding drought tolerance crops. The genetics part, of course, I'm a geneticist. I'm concerned how to improve the genetic performance by enabling the genes to be more efficient in producing more food.
Professor, for that insightful presentation.
Directing this interesting talk and answers the questions and answers section of this talk.
Okay, doctor, there are so many questions that people have for you. I don't know how I would send these questions to you, but I encourage every one of us, if at all you have these burning questions, please reach out to the doctor. That's in his account, that's the NileWell account. Doctor will definitely get your email and answer your questions. For now, friends, I don't want to waste so much time. I want to say you have been very good guests, and we hope we can have you the next time we have these science cafes. Otherwise, remember, this has been live on YouTube, but also remember to open up your account on NileWell so that you can get all these goodies coming to you. Otherwise, I've been your host for today, Andrew Aijuka, standing in for Delicate. Ogada. And till next time,
See you.
bye-bye.
Bye.